All right, let's continue with our discussion of metabolism, but let's now focus on the enzymes, which actually help in the whole process of metabolism. So you know that enzymes are proteins. We've talked about those before, and that this idea that they are catalysts. So a catalyst is something that is able to speed up a reaction without really entering into the reaction at all. So the enzymes are basically helping a reaction to happen, but they aren't a part of the reaction. So what they do in particular is enzymes are able to lower the activation energy barrier. So most chemical reactions, as we said before, will proceed over a period of time and then you get a product. But in some cases, you need an enzyme to facilitate that because it would take either forever for that reaction to take place or it'd be very unlikely because there's too much of a barrier to those substrates actually engaging with each other and forming the products. So what enzymes do is they bind to the reactants. And then what they do in binding is they provide sort of a place, if you want to think of it that way, for some of these reactions to take place. But they also do something else which is interesting, and that is that part of the way that they catalyze a reaction is by causing some strain on the reactants or putting them in a position so that they can react better. So let's take a look. Oh, actually, before I do, I wanted to note what they said here on the slide, that as you know, enzymes are very specific to what kind of a reaction that they catalyze. So when we looked at the enzyme lactase, remember ASE is very often an ending for enzymes, we know that it operated specifically on a substrate we call lactose, which is milk sugar. So thinking about this, it's kind of an important piece. So lots of enzymes for all sorts of different reactions. So let's look at the next one. So this idea of that the enzyme works with a very particular substrate, we say that it is enzyme substrate specificity. So it just means it's very specific to some substrate. So when we look at the enzyme, remember that all of these molecules are three-dimensional, and that reactants are the things we are calling substrates. So that when an enzyme is formed, it's a protein, right? So it's got to do all the folding. And when an enzyme does its final tertiary folding, or even quaternary in some cases, what you're going to get as a consequence of that very specific folding is you're going to have an enzyme. So here's the enzyme down here represented, that will have this active site, which is the area that it's actually going to be able to pick up the thing it's working on, its substrate. So, for example, if this happened to be the enzyme lactase, and this happened to be milk sugar or lactose, notice that they would fit beautifully together. They would form a complex. The enzyme actually changes shape a little bit as it sort of snuggles into this active site, and then that helps with this little bit of strain on this substrate is going to help with ultimately creating the final thing, which is the products. The products are going to leave the active site, and then this enzyme is free to go do that again with another substrate. So here's just a three-dimensional view of what some enzymes would look after. Remember, these would all be amino acids, and they would all have done all of their folding, and here is the active site of this particular enzyme, where the blue is representing the place where it would bind to that substrate. And then right here, this red in here is called the catalytic site. So this is the part that's going to help do something that is going to bring down that activation energy barrier. Now, what this is called, this little change in the shape of this protein that makes this reaction proceed forward, is called an induced fit. So this is actually a model, kind of like we've seen with Pogles. The induced fit model basically explains this idea that once the substrate enters the active site of the enzyme, that causes a slight conformational change, a slight change in the shape of the enzyme, which is going to help for this idea of breaking down, in this case, this substrate into these two products. Here's another example. You can see this by the image. So here's in this case, we have two substrates that are going to go into this active site of this enzyme, and I'm going to create a product then that would be, in this case, we're going to look at an anabolic reaction. In other words, we're taking smaller things, we're building something bigger. This is to also remind us, remember, proteins, we're talking about enzymes are proteins, are an example of proteins, and that the shape of that enzyme is going to be determined by the order of the amino acids in the primary structure of the protein, and then how it eventually folds due to that order of those amino acids. Now, things which can interfere with the structure of a protein, if you remember, would be things that might be in the cellular environment, like if the temperature, it becomes less optimal, in other words, it's not normal temperature, or maybe the pH changes. Those can affect the way the enzyme operates. So here's more on how the enzymes work. So once the enzyme has the substrates, has them in that active site, now it's going to provide a special environment for that reaction to take place. So it could be that that active site might have something that is related to it having some acidity or having some charge that's going to help the substrate to bind and to have the reaction. It can cause, remember I had said it can like stress the substrate, like put a little strain on it so it's less stable. Can cause an intermediate kind of reaction to take place. In other words, chemically change it a little bit, making that substrate less stable again. So then once that all happens, then the product gets released and the enzyme goes and does its job somewhere else. Now, this is from your text, so I encourage you to go look at this. This is a nice link that they've provided you to see how enzymes actually catalyze reactions. So very useful to use the links in OpenStax. Now, last, enzymes have to be regulated. Enzymes need to have certain things which are going to influence whether or not they are going to do a job or perhaps not. So we already said that enzyme shape, because they're proteins, can be regulated by conditions around them, such as a change in temperature or pH. But there also can be some things which will actually inhibit or promote an enzyme's ability to work. And some of these things are called coenzymes or cofactors. Now, you actually know some of them because some of them include some of the vitamins that we take, or maybe there'd be even minerals that we take, so that'll be somehow helping an enzyme to work. Or perhaps there'd be something else. So let's take a look at this. So we can inhibit an enzyme. We can't, but I mean, there are things, chemicals or substances, which can go in and either go into the active site where that enzyme would normally be, and therefore kind of like sort of put the brakes on it for a while. That means that would be a competitive inhibitor, something that actually fits in that same active site as the substrate, and it's going to stop the activity of the enzyme. The other possible way it can be inhibited is called a non-competitive inhibitor. And what that is it goes into another area, not the active site, but another area of the enzyme. Kind of changes its shape a little bit, which then is going to interfere with that enzyme performing until that inhibitor is removed. So this isn't the best picture. I'll show you some other ones in class. But what happens is when you have an inhibitor, in other words, something that's going to go into the enzyme, either in its active site or its non-active site, and going to cause something to slow down or to affect that enzyme's ability to do its job, what you see is this. So on a graph, if this would be the normal enzyme here in purple, this, in other words, here's the rate of reaction. So the reaction rate is increasing here. And here it has to do with how much of the substrate is available, or substrate concentration. When you add an inhibitor, this would be its normal activity. At some point, the enzyme just sort of can't act any faster. So you see that that curve flattens out. That would be where the enzyme actually would be called saturated, meaning that there's not enough enzyme available for the amount of substrate. It can't go any faster. But here's the other two possibilities. So if you put in a competitive inhibitor, something that interferes in the active site, the enzyme can still work, but it's much less effective. Its rate of reaction is slower. It takes longer to reach this sort of maximum level. Whereas a non-competitive inhibitor, which actually changes the shape of the enzyme, it might allow for some enzyme that's in the environment to work, but it keeps interfering. So what you see here is a sort of a much lower rate of reaction. Now, these are some terms too, and I want to get into these a little bit later because I think this is a concept that we're going to see. But I think that's a sufficient little dose of enzymes and how enzymes can be regulated through one process called inhibition. So how do we either competitively inhibit or non-competitively inhibit an enzyme's ability to do its job and to cause to create the products of that reaction. So we'll stop right there.